Welcome to our deep dive into the world of molecular biology. And you know the story of DNA. You've given us some really fascinating excerpts from Horace Judson's book, The Eighth Day of Creation,
Yeah,
and it's incredible.
it's a great book.
It's a really incredible journey through all the key discoveries and the people who revolutionized how we understand life.
Yeah, and it's more than just the structure of DNA.
Yeah.
Judson really takes us back to the very beginnings of molecular biology,
Right.
a time when, scientists were really just starting to grapple with this whole idea of biological specificity.
So back then, I mean, they didn't even use the term molecular biology.
Oh.
So how did they even approach something as complex as,
Yeah,
how molecules recognize each other and interact with each other?
Yeah.
It seems like a huge leap.
you're absolutely right. It was a huge leap. And, one of the pioneers leading this charge was Max Delbruck.
Okay.
And this is where it's really interesting because
Yeah.
he was a physicist, but he decided to sort of dive headfirst into the world of biology
Mm.
and specifically focusing on these things called bacteriophages, which are viruses that infect bacteria.
So he went from studying the universe to studying these tiny viruses.
Exactly.
What sparks that kind of change?
He had this belief that by studying life at its most basic level,
Okay.
using these phages,
Uh-huh.
we could uncover, the fundamental principles that apply to all living things.
Right.
So it was a very bold approach. And actually, at the
Yeah.
time,
Yeah.
some scientists thought it was a bit of a step down to be studying these phages.
Really?
Yeah,
Well, he certainly proved them wrong.
he did.
But it wasn't just his research that had such a big impact, was it? I mean, he also had this knack for mentoring future stars in the field.
Oh, absolutely.
Hmm.
He fostered this really incredible environment, of intellectual curiosity and debate,
Hmm.
much like the atmosphere he experienced, with Niels Bohr,
Oh, wow.
a famous physicist.
Yeah.
And he mentored some really brilliant minds, including Salvador Luria,
Right.
even James Watson,
Wow.
who, as you know, would go on to play a major role in the whole DNA story.
Talk about a scientific family tree.
Yeah, exactly.
So while Delbruck and his team were busy with their phages,
Yeah,
other scientists were tackling this mystery of DNA itself.
that's right. There was a scientist named William Astbury,
Okay.
and he was using this technique called X-ray diffraction to try to study the structure of DNA.
Yeah.
And his work was really groundbreaking.
Okay.
I mean, his images suggested that there was this regular repeating pattern in the molecule.
Wow.
And he even proposed that the bases were stacked like coins.
So he was already getting these first glimpses into the structural secrets hidden within DNA?
Yes, absolutely.
Yeah.
His work was essential.
Okay.
But to really crack the code of DNA, someone needed to have this deep understanding of chemistry, physics,
Right.
and kind of a touch of model building genius.
Okay.
And that's where Linus Pauling enters the picture.
Ah, Linus Pauling, the master of chemical bonds.
Yes.
I mean, his work completely changed the way we think about how atoms connect to form molecules.
Absolutely.
It's like he laid the foundation for everything we know about molecular structure.
Exactly. And his approach to solving molecular structures was really innovative.
Okay.
Take his work on the alpha helix, for example,
Uh-huh.
which is this key structural element in proteins.
Right.
He actually built models
Wow.
with cutouts of amino acids,
Uh-huh.
trying to figure out how they could fit together based on the chemical principles.
That's amazing.
So it's kind of like solving this giant 3D jigsaw puzzle,
Yeah.
but instead of having a picture, you're creating a molecule.
That's such a great analogy.
Yeah.
So this model building approach must have been really influential in the race to discover the structure of DNA.
Oh, absolutely. In
Yeah.
fact, Pauling himself actually tried to solve the structure of DNA
Oh, okay.
and even proposed a triple helix model.
Wait, he got it wrong? How did that happen?
Well, he didn't have access to the most accurate data at the time.
Ah, okay.
And this is a really important point, I think, for anyone trying to understand how science really works.
Yeah.
Collaboration and access to good data are essential for breakthroughs.
Right?
And Pauling's misstep really highlights that.
Okay, so while Pauling was working on his triple helix model,
Yeah.
the race to DNA was really heating up in other labs,
Right.
specifically at the Cavendish Laboratory in Cambridge.
Exactly.
Gotcha.
That's where we meet James Watson and Francis Crick.
Okay.
These two scientists with just this insatiable curiosity and a real knack for out-of-the-box thinking.
Mm-hmm.
They were fascinated by this DNA puzzle
Right.
and eager to sort of piece together all these different clues.
But they weren't the only ones on the case, right?
No.
Because over at King's College London, you had Maurice Wilkins and Rosalind Franklin,
Yes.
who are also working diligently on DNA using that same X-ray diffraction technique.
Right, and this is where the story takes a bit of a dramatic turn. Because the relationship between Wilkins and Franklin was, shall we say, less than ideal.
Yeah.
There was a lot of tension, miscommunication, and a real clash of personalities.
It's a good reminder that science isn't just about data and experiments, right?
Right.
It's also about the people involved.
Yeah.
And how their relationships can affect the whole course of discovery.
You hit the nail on the head.
Mm.
Despite these challenges, though,
Okay.
Franklin made a crucial breakthrough.
Okay.
She discovered that DNA could exist in two forms.
Huh.
There was this crystalline form that produced these really clear diffraction patterns,
Okay.
and then a wet form that was more hydrated. And her meticulous data collection, was really invaluable.
And then there's this famous image, Photo 51,
Yeah.
taken by Franklin and her student Raymond Gosling.
Right.
And this is considered one of the most important images in the history of science, isn't it?
Absolutely.
So what made it so crucial?
Well, that image, along with Franklin's really precise measurements,
Sure.
it provided the missing piece of the puzzle.
Okay.
It really revealed the helical structure of DNA,
Mm-hmm.
which was crucial for Watson and Crick to be able to build their accurate model.
Right.
So they combined their own insights with all this existing data from Pauline Chargaff Wilkins
Wow.
and, of course, Franklin.
So it was really this culmination of efforts,
Mhm.
a scientific relay race,
Exactly.
where each scientist contributed this crucial piece of information.
Yeah, and their model of this double helix with its elegant base pairing
Right.
and the implications for genetic information, it was a monumental achievement.
It was.
It was the key that really unlocked this whole new world of understanding about life.
But that was just the beginning, right?
It was just the
I
beginning.
mean, the discovery of this double helix led to a whole new set of questions.
Oh, yeah.
About how DNA actually works.
Exactly. How does DNA replicate itself?
Right.
How does it guide the creation of proteins? These questions really fueled this explosion of research in this burgeoning field of molecular biology.
It's amazing how one discovery can lead to so many more questions and propel science forward.
Absolutely.
Yeah.
And the story of how scientists tackled those questions is just as fascinating as the discovery of DNA itself.
I bet.
Yeah,
And that's what we're going to dive into next time.
we will.
All right, sounds good.
Yeah.
So last time we left off with this incredible discovery of the DNA double helix, this monumental achievement.
Yeah.
But, as you mentioned, it also opened up a whole new set of questions.
It did.
I mean, how does DNA actually make copies of itself? And
Yeah.
how does it direct the creation of proteins,
Yeah.
these workhorses of the cell?
Yeah, those are really the big questions that drove the next wave of research in molecular biology.
Okay.
And as scientists dug deeper, they uncovered this whole new layer of complexity.
Okay.
The central dogma of molecular biology.
Okay. I've heard that term before, the central dogma. But to be honest, it sounds a little intimidating.
Right.
What does it actually mean?
And it's actually a pretty simple concept.
Okay.
At its core, it describes the flow of genetic information
Okay.
from DNA to RNA to protein.
Okay.
So DNA holds the blueprint,
Uh-huh.
the genetic code.
Right.
That code is transcribed into RNA,
Okay.
which acts as a messenger,
Uh-huh.
carrying the instructions to the ribosomes,
Right.
which are the protein-making factories of the cell.
Okay.
And finally, those instructions are translated
Okay.
into the specific sequence of amino acids
Uh-huh.
that make up a protein.
So it's like a chain of command,
Yeah,
this molecular relay race,
exactly.
where each molecule has a specific role
Uh-huh.
in passing on the genetic instructions.
Exactly.
Okay, so for a while, scientists thought that RNA was just this kind of passive messenger, this simple go-between
Right.
for DNA and proteins.
Yeah, but as they explored further,
Mm-hmm.
they realized that RNA was actually much more complex and versatile.
Hold on, RNA is more than just a messenger.
Yeah,
Okay, I'm intrigued.
it is.
What kind of hidden talents does RNA have?
Well, one of the first hints actually came from studying viruses.
Okay.
Some viruses, like the tobacco mosaic virus,
Mm.
they don't even have DNA.
Really?
Yeah, their genetic material is made entirely of RNA.
So these RNA viruses are basically breaking the central dogma.
In a way, yes.
Wow.
They are. I mean, these viruses showed that RNA could actually store genetic information,
Ah.
replicate itself,
Wow.
and even direct the synthesis of proteins,
Okay.
all on its own, without any help from DNA.
So RNA can be like the star of the show,
Exactly.
not just a supporting actor.
It's not just a bit player.
That's incredible.
And that realization sparked this whole cascade of discoveries.
Okay.
Scientists started finding all these different types of RNA,
Uh-huh.
each with its own specialized function.
Okay.
So there's messenger RNA or mRNA,
Right.
which carries the genetic instructions from the DNA.
Okay.
Then there's transfer RNA or tRNA,
Oh. Mm-hmm.
which acts like this molecular interpreter,
Mm.
translating the language of nucleic acids into the language of proteins.
Okay.
And then there's ribosomal RNA or rRNA,
Okay.
which is a key component of the ribosomes themselves.
Wow. So many different types of RNA,
Yeah,
each with its own important job to do.
it is pretty amazing.
It seems like RNA is involved in every step of this protein synthesis process.
You got it. And this discovery really changed how scientists viewed RNA.
Right.
It wasn't just this passive messenger anymore.
Uh-huh.
It was an active participant in this whole process of creating proteins,
Wow.
almost like a stage manager,
Yeah.
coordinating the whole production.
Okay, so this is where things get even more interesting, right?
They do.
Because you mentioned that RNA can actually act as an enzyme.
Yeah.
And I thought that was supposed to be like the exclusive job of proteins.
Right. That was a huge surprise.
Yeah.
The discovery of these RNA enzymes or ribozymes
Mm-hmm.
was a real game changer.
Okay.
It really challenged this traditional view of RNA
Right.
and added this whole new layer of complexity to this central dogma.
So RNA can be a messenger, an interpreter, a
Yeah.
structural component,
Uh-huh.
and even an enzyme that speeds up these chemical reactions.
Exactly. It's amazing.
It is, isn't it?
Yeah.
And the story of RNA is far from over.
Right.
Scientists are still uncovering, new types of RNA,
Yeah.
new functions,
Yeah.
like small RNAs that regulate gene expression,
Okay.
and long non-coding RNAs with these kind of mysterious roles.
Hmm.
Some scientists even believe that RNA might have been, the key player in the origin of life itself.
Wow. That's a mind-blowing thought.
It is.
Okay. So while all this research on RNA was going on,
Yeah,
scientists were also trying to decipher the genetic code itself.
right.
I mean, we know that DNA carries the instructions for building proteins.
Uh-huh.
But how do you translate the language of DNA into the language of proteins?
Right.
That seems like an incredibly complex puzzle
It was, and it involves some really brilliant minds.
and some very clever experiments. One of the early pioneers in this area was George Gamow,
Okay.
the physicist who actually proposed the Big Bang Theory.
Wow. Okay.
He got interested in the genetic code and came up with this idea called the Diamond Code.
Okay.
It turned out to be incorrect,
Oh.
but his work really sparked further research
Wow.
and inspired others to tackle this problem.
Yeah. So Gamma, the Big Bang guy, gets intrigued by the mysteries of DNA.
He does.
That's a testament to the power of scientific curiosity.
It is.
But how do they actually crack the code? It seems like you'd need like a molecular Rosetta Stone or something.
Right. Well, Francis Crick, always a visionary,
Mm-hmm.
he came up with this idea called the adapter hypothesis.
Okay.
He proposed that there had to be some kind of molecule that acted as a bridge
Okay.
between the RNA and the amino acids that make up proteins.
So these adapters would be like these little molecular couriers
Exactly.
carrying the amino acids
Yeah.
to the right spot on the RNA template.
Exactly. And, his hypothesis was initially met with some skepticism,
Okay.
but it turned out to be right on the mark.
Wow.
And it paved the way for the groundbreaking work of Marshall Nirenberg and Heinrich Matthaei,
Okay.
who finally cracked the code.
Okay.
They actually figured out the relationship between specific RNA sequences
Mm-hmm.
called codons
Okay.
and the amino acids they specify.
So they basically created a dictionary
Yeah,
that translates the language of DNA into the language of proteins.
exactly. That must have been an amazing moment in scientific history.
It must have been.
Yeah, it was a huge breakthrough,
Uh-huh.
and it opened up this whole new understanding
Uh-huh.
of how genetic information is expressed.
Right.
But the story doesn't stop there.
Of course not.
Now that scientists understood the code,
Yeah.
they could turn their attention to this amazing world of proteins.
Right, because proteins are the real workhorses of the cell, right?
They are.
I mean, they do everything.
They do.
From catalyzing reactions to transporting molecules to providing structural support.
Right.
But how do they achieve such diversity and specificity in their functions?
Well, it all comes down to their three-dimensional structures.
Okay.
Remember Linus Pauling and his work on the alpha helix?
Yes, of course.
He was the one who figured out that proteins fold into these specific shapes,
Right.
like a piece of origami.
Right, like a piece of origami. But how do those shapes actually determine what a protein can do?
Well, you can think of it like this.
Okay.
Each protein has a unique surface
Okay.
with specific bumps, grooves, and charges,
Uh-huh.
all determined by its folded shape.
Okay.
And these features allow proteins to interact with other molecules
Uh-huh.
in these very specific ways, like a lock and key.
So the shape of a protein basically dictates who it can bind to and
Yeah.
what it can do.
Exactly. It's like a molecular dance,
Right.
where each protein has its own unique moves.
It really is like a molecular dance.
It is.
And as scientists started solving the structures of more and more proteins,
Yeah,
they started noticing some interesting patterns.
they did.
Yeah.
Certain structural motifs, like the alpha helix and the beta sheet,
Mm-hmm.
kept appearing in different proteins,
Okay.
even those with totally different functions.
So it's like nature has this limited set of building blocks that it uses to create this huge variety of protein structures,
Exactly.
like playing with molecular Lego.
It is. And that modularity is incredibly important for evolution.
Okay.
New proteins can arise not only from these gradual changes in DNA sequence,
Okay.
but also from shuffling and recombining these existing structural modules
Right.
called domains.
Okay.
So it's a really powerful way to create diversity and complexity.
So protein evolution is this kind of mix of fine-tuning and these bold rearrangements. It's almost like a choreographer trying out new moves
Right.
and then incorporating the best ones into a new dance routine.
Exactly. A perfect description.
So this understanding of protein structure and function
Yeah.
has really opened up all these new avenues of research.
It has.
Yeah.
Scientists are working on predicting how proteins fold,
Uh-huh.
understanding how they interact
Mm.
to form these complex molecular machines,
Right.
and even designing new proteins with specific functions.
It's mind-boggling to think that we can now visualize these incredibly intricate molecular structures
It is.
and start to understand how they orchestrate this whole symphony of life.
Yeah.
But there's one crucial aspect that we haven't talked about yet.
Right.
How is all this amazing activity controlled within the cell?
Right.
I mean, how do cells make sure that the right proteins are produced
Yeah.
at the right time and in the right amounts?
That's where we enter this fascinating world of gene regulation,
Okay.
the control center of the cell.
Wow. It's been quite a journey through this molecular world,
Yeah.
from the discovery of DNA to this intricate dance of proteins.
It has.
But as you just mentioned, there's this whole other layer of complexity
Yeah.
in regulation.
Yes.
How do cells manage this incredible orchestra of making sure that the right proteins are produced at the right time
Sure.
and the right amounts?
Well, it really is like, having a conductor leading a symphony,
Okay.
making sure that each instrument plays its part harmoniously.
Right.
Without gene regulation, it would just be chaos,
Uh-huh.
a jumble of proteins
Yeah.
leading to dysfunction and even disease.
So how does this conductor, this
Hmm.
gene regulation system actually work? I mean,
Yeah.
it just seems impossibly intricate.
It's definitely complex,
Yeah.
but it's also remarkably elegant.
Okay.
And some of the first clues about how it works actually came from studying bacteria.
Okay.
These tiny microbes
Huh.
that have taught us so much about the fundamentals of life.
Ah, bacteria.
Back to the basics.
Yeah.
I'm starting to see why they're like the unsung heroes of molecular biology.
They are. Their simplicity made them the perfect model system for unraveling these mysteries of gene regulation.
Remember Jacques Monod and Francois Jacob,
Yeah.
who deciphered the lac operon?
Of course. They showed how these genes involved in lactose metabolism in E. coli
Right.
were all bundled together
Yeah,
and controlled by this single switch,
right.
the operator.
Exactly. It was like a mini gene regulatory circuit,
Yeah.
turning on or off,
Uh-huh.
depending on whether lactose was present or not.
So elegant.
It is. And that operon model, with its repressors and inducers, it provided this framework for understanding
Okay.
how genes could be switched on and off in this coordinated way.
Okay.
But as scientists dug deeper,
Yeah.
they found that bacteria actually had this whole toolkit of regulatory tricks up their sleeves.
Really? What other kinds of tricks are we talking about?
Well, they discovered systems that could amplify gene expression,
Huh.
creating these bursts of protein synthesis on demand.
Wow.
They found feedback loops where a gene's product could actually control its own expression,
Okay.
like a self-regulating thermostat. And they uncovered these global regulatory networks
Okay.
where master switches controlled multiple genes,
Wow.
orchestrating these complex responses to environmental changes.
So even bacteria, with their seemingly simple setup,
Yeah.
have this sophisticated system for fine-tuning gene expression
They do.
and responding to their surroundings.
Absolutely.
Yeah.
But as scientists turn their attention to more complex cells,
Right.
those with a nucleus
Uh-huh.
and all sorts of specialized compartments,
Yeah.
the story of gene regulation got even more intricate.
I can imagine eukaryotic cells with their, really intricate organization
Right.
must have a much more elaborate system for controlling their genes.
You're right. In these cells,
Yeah.
DNA is packaged into these things called chromosomes,
Uh-huh.
which are incredibly complex structures.
Right.
And this packaging itself actually plays a role in regulating gene expression.
Okay.
So it's not just about repressors and operators anymore.
Uh-huh.
The physical structure of the DNA matters too.
So it's not just a matter of turning genes on or off.
Right.
It's also about how accessible those genes are in the first place.
Exactly. Imagine, a library with some books locked away in a vault
Okay.
and others, neatly arranged on shelves ready to be read.
Yeah.
The structure of the chromosome, called chromatin,
Uh-huh.
determines which genes are open for reading
Right.
and which ones are kind of tucked away.
That's a great analogy.
Yeah,
But this chromatin structure isn't static, is it? I mean, can it change?
it absolutely can.
Okay.
Enzymes can modify the chromatin,
Uh-huh.
making certain genes more or less accessible.
Huh.
So it adds another layer of control
Okay.
for gene regulation.
So it's like having these librarians constantly rearranging the books,
Right.
making sure that the right ones are available at the right time.
Exactly.
Yeah.
Perfect way to put it.
So this dynamic interplay between DNA sequence, chromatin structure, and all these regulatory proteins
Yeah,
creates this incredibly finely tuned system
it does.
for controlling gene expression in eukaryotic cells.
It does.
It's mind-boggling to think about all these levels of control working together.
It is.
But I can see how this complexity allows for the incredible diversity we see in these eukaryotic organisms.
Right.
I mean, every cell in our body has the same DNA,
Right.
but they obviously do very different things.
Exactly. You've hit on a key point.
Okay.
It's this intricate system of gene regulation
Yeah.
that allows for cell specialization.
Okay.
A neuron, a muscle cell, a skin cell,
Huh?
they all have the same genetic blueprint,
Right.
but they express different subsets of genes,
Okay.
giving them their unique characteristics.
So gene regulation is essentially what orchestrates the development of a multicellular organism
Yes.
from a single fertilized egg.
It is. That's incredible.
It's pretty amazing.
Oh.
And it's also, what allows cells to respond to environmental cues,
Right.
adapt to changing conditions,
Uh-huh.
and maintain this delicate balance we call life.
So we've got these protein-coding genes.
And all these intricate regulatory mechanisms that control them.
Yeah.
Is there anything else going on in the genome?
Well, this is where things get really interesting again.
Okay.
Remember how we said that for a long time?
Yeah.
Scientists thought that RNA was just this simple messenger.
Yeah.
Turns out that's just the tip of the iceberg. We now know that most of the RNA in a cell
Mm-hmm.
doesn't actually code for proteins.
Wait, so all that RNA was just hanging out doing nothing?
That's what scientists initially thought.
Wow.
They called it junk DNA.
Right. Junk DNA.
But, as usual, nature is full of surprises.
Yeah.
It turns out that these non-coding RNAs
Okay.
actually play this vital role in gene regulation.
So junk turned out to be treasure.
It did.
What kind of roles do these non-coding RNAs play?
Well, there's a whole world of them.
Okay.
Some are tiny, like microRNAs and siRNAs,
Okay.
that can bind to messenger RNAs,
Mm-hmm.
blocking their translation into proteins,
Okay.
or even targeting them for destruction.
Wow.
So it's like having this team of molecular sensors
Right.
silencing specific messages before they can be expressed.
Okay, what about the longer non-coding RNAs?
Those have some really fascinating
Okay.
and often mysterious functions.
Uh-huh.
Some act as scaffolds,
Okay.
bringing together different proteins involved in gene regulation.
Mhm.
Others act as decoys,
Okay.
binding to regulatory proteins and
Mhm.
preventing them from interacting with their target genes.
So it's like a whole new level of complexity in this gene regulation game,
It is.
with all these non-coding RNAs playing all these different roles,
Yeah.
influencing which genes get expressed and when.
It's the symphony of regulation,
Yeah.
with multiple layers of control,
Right.
ensuring that the right genes are expressed at the right time
Uh-huh.
and in the right amounts.
Wow. That was an incredible journey.
It was.
From the early days of phage research to deciphering the genetic code,
Yeah.
unraveling the complexities of protein structure and function, and now diving into this intricate world of gene regulation. Well, we've covered an amazing amount of ground.
We have. And through it all, we've seen how all these discoveries, intertwine
Right.
to paint this picture of the molecular machinery that drives life.
Yeah.
This intricate dance of molecules working together in harmony.
It really is.
Yeah.
It's both humbling and exhilarating to realize how much we've learned
It is.
and how much there's still to explore.
Yeah.
What new questions does this deep dive spark for you?
Well, for me, it's the realization that the more we learn,
Yeah.
the more questions we uncover.
Wow.
The field of molecular biology is constantly evolving.
It is.
And I'm really excited to see, what new discoveries lie ahead.
Yeah.
Where will this knowledge take us next?
Right.
What new mysteries will we unravel?
That's the beauty of science, isn't it? This never-ending quest for knowledge,
It is.
driven by curiosity and wonder.
Absolutely.
And for anyone listening, if this deep dive has sparked your own sense of wonder, there's a whole universe of knowledge waiting to be explored.
There is.
That be exploring.